First of all, I'm very happy to see you. Thank you for being here. And now, I want to give a brief introduction of our guest, Christian Rosenmund from Charité Medical University. I think it's in Berlin. And briefly, Professor Christian Rosenmund received his PhD in physiology at the Valley Institute of Oregon Health Sciences University, and then he became professor since 2009 at the neurobiology of the Institute of Neurophysiology at the Charité in Berlin, which is a part of NeuroCure cluster of excellence. And previously, he was a professor at the Baylor College of Medicine in Houston, and he was also a university lecturer in physiology at the University of Göttingen from 1999 to 2003. And actually, I remember when I was a PhD student in Freiburg, I think it's from 1998 to 2004. If you remember, maybe you visited Peter, and I was a PhD student. Mm-hmm. So I'm very impressed with your talk, and I remember I met you because at that time, Peter asked me to work on Mons-Survival Bouton and to study semantic transmission. Yeah. And I was really very reluctant to do that. So finally I said, no, I don't want to work on semantic transmission because it's very, very difficult for me. And a lot of membrane biophysics, a lot of equation. Probably I'm not good at mathematics. And also Peter was very good in doing this modeling, and also Evinair, they are really big guys. And so, and also on the other side, I'm really very afraid of all these complex, snare complex, and all these semantic proteins. So if you know, when I give a talk about semantic transmission, I always skip all these complex, these semantic complex like monk, rab, snare complex, Syntaxin, blah blah blah, a lot of semantic protein, and also of course the structure is also very complicated. So that's why I didn't continue to work on semantic transmission. Okay, so and Christian has been a big guy at this field. And he has been persistently working on semantic transmission since he was a PhD student with Steve, right?
No, Gary Westbrook was my PhD.
You worked with Gary Westbrook.
Yeah, as a PhD, and then it was
Gary very well.
Yeah.
He was also here several years ago. Oh, Gary. Yeah. And then you worked with another big
Chuck
guy.
Stevens. Yeah,
Chuck Stevens.
Yeah.
Chuck Stevens is really a very big guy. If you know, in my office there is a textbook. I think it's written by Chuck Steve and maybe Evinair. It's called Synapse. Yeah. Okay. So now you know. Today we have a really big expert in semantic transmission. And without further ado, let's welcome Professor Christian Rosenmund.
Thank you. Thank you so much. Thank you for making possible for me to visit here. It's always exciting to learn about the microphone needs to be turned on, I guess, or so. Okay.
Or let's use this.
Okay. Yeah. Okay. Yes. So, I guess. Okay, then you hear me better this way. No, it's very exciting for me to see different environments, and it was really kind of a short notice visit here, which I'm very glad that you could make this happening, and I'm gladly tell you a little bit about the work we're doing. Of course, you introduced me already very well. You know, my lab has been working on fundamental physiology of synaptic transmission. In particular, we are interested in presynaptic neurotransmitter release. And so we are interested in these little red dots here that you see. And this is a neuron here in culture. This is a system that we mainly work. And they, of course, use synapses to communicate with each other. And just as a brief introduction, to start simple, you know, we have the presynaptic nerve terminal, the postsynaptic side. The presynaptic side is, the job is to transduce the incoming action potential into a fusion of a synaptic vesicle. And the trigger for that is the influx of calcium, which of course is caused by the action potential. The action potential induces the opening of calcium channels, and then within 50 microseconds or so, the synaptic vesicle fuses with the plasma membrane. And this is probably one of the fastest cell biological processes that we know of, this triggering of vesicle fusion by calcium. And that has been fascinated me throughout my career, that you have this very rapid action potential formation in the neuron, the excitability, and then you have the synapse that has to be just as fast as a cell biological apparatus to the ion channels and the membrane of neurons. And so then it releases a neurotransmitter in the cleft, in the central nervous system it's glutamate or GABA mainly, and then you have the resurrection of an electrical signal at the postsynapse by opening of ligand-gated ion channels. So again, this happens very rapidly, and one of the reasons why you worked on the mossy fiber synapse, which is really the hardest synapse to work on, the synapse we work on so simple, right? I can teach you in one day how to do this. So it's it's not very difficult. But the mossy fiber is like the God synapse, okay? And so don't be humble about it. It's really a very special and difficult prep to work with, indeed. And so I've been interested in how actually does the calcium triggers the fusion process so efficiently and so rapidly. What are the mechanisms, what is the machinery underlying this process? And of course, there are models now that have been developed over the years, knowing about the proteins, using genetics, knockout of mice and removing proteins, and then you start seeing some phenotypes, and then you're essentially building a model how the whole process works. These are still often cartoons that we're using, like these, like how they could work. But we understand, we still, after working on this for 25 years or so now, we still a lot of open questions. And even the very fundamental apparatus of the fusion apparatus is a snare complex that is clearly very important for the vesicle fusion process. So we know the vesicle of the synaptic vesicle has to fuse with the plasma membrane. And that's difficult because the membranes are negatively charged phospholipid membranes, so they have expelling, they don't like to come close to each other. And yet you have to overcome that energy barrier with the calcium trigger. And so therefore you need, for example, the snares that are embedded in the two different membranes, synaptobrevin in the vesicle membrane and Syntaxin on the plasma membrane. Also you have SNAP25 here as a third protein. And they form this kind of coiled structure that sort of twists this vesicle closer to the membrane. And that's kind of the trick is, you will see, this is kind of the theme that we find, you have to get the vesicle membrane close to the plasma membrane, and the closer the better. This is kind of a key issue. That's why the analysis of electron microscopy has been very, very important rather recently to understand the entire fusion process, because distance is extremely important. So the snare complex is in a machinery that allows it to bring those two membranes very close. And after the fusion, you have the snare complex being in the so-called cis configuration. Here it's in the trans configuration. And they have been really instrumental for understanding the fusion process. And I will focus today in particular about Syntaxin 1A as playing in multiple roles in this kind of cycle that I've shown you here, in the docking, in the priming, and the calcium triggering, and how it affects actually different types of fusion. There's a calcium triggered fusion, and there's also spontaneous fusion. So vesicles, once they become fusion competent, they can also without calcium fuse. And so we are looking at these kind of properties in more detail. So this is just a reminder for you. So, please ask a question if you don't know anything. you interrupt me, please, if there's something unclear. But so this question of docking and priming is very important in understanding how fusion happens. So, and mainly what have the view of the central synapse, and we use electron microscopy to see if vesicles attach or dock to the vesicle membrane. And this is an important step because it obviously brings the vesicle and the plasma membrane very close. Priming, on the other hand, is a functional definition. It means a vesicle is primed, it is ready to fuse when the trigger comes. Okay, one is a morphological definition, it's a docking, and the priming is a functional definition. Okay, and some of the ideas is that, of course, docking and priming has to sort of both happen, that the vesicle go on, and but not necessarily they are always a docked vesicle is not necessarily a primed vesicle. So that's what I want to show you at first. First, I show you my model system that we love to use. It's the most simple circuit you can think of. It's a single neuron on an island, micro island growing here, and essentially the neuron makes synapses and axons and dendrites and synapses and sort of connects onto itself. And then we use an electrode on the soma and then depolarize and use an action potential in the axon. And then a few milliseconds later, you have a synaptic response that we can very nicely quantify. The beauty of that system is that we know exactly the input and the output in this circuit, right? Because if we trigger an action potential, we measure the output from these 200 or so synapses, and we can exactly quantify how efficient is release happening. And this is very important in studying, for example, mutants. You want to know, is this mutant affecting release or not? Is it affecting docking? Is it affecting priming? What kind of property does it affect? And here we have a very nice system where we can be very quantitative about release. And that's what is the strength of the system. It's artificial, but it has a strength in being very, very quantitative, and therefore very powerful in analyzing molecular mechanisms, molecular machines. And that's what I sort of built my career on, using this sort of bottom-up approach. There is another way of stimulating release beside action potential is by using hypertonic solutions. So you increase osmolarity of your solution for like a few seconds, and that forces all these green vesicles are primed fusion-competent vesicles. With this hypertonic pulse, you force them to be released, and then you get this inward current, essentially a barrage of spontaneous single vesicle fusion events that occur, and then you can take this transient component of the response here and sort of measure how many fusion-competent vesicles are actually made from these 200 synapses. And then you can say, okay, this cell has a pool of 6,000 synaptic vesicles that are fusion-competent distributed along 200 synapses, and so you can see that among those 6,000 vesicles, 300 are released by an action potential. You can really calculate release probability and all these things very quantitatively. You can look also at spontaneous release, which I mentioned to you is the other form of release, and that is also the measure of this release is also a measure of the activity of the release apparatus itself, and is also under intense debate what the role of this activity is in neuronal function and in circuits. Okay, so what I'm going to tell you is an approach that we use to study Syntaxin in release in the multiple steps. And it's a classical approach that we do in the lab. We take a knockout mouse for the gene of interest that we're interested in, Syntaxin. There are two genes, Syntaxin 1A and 1B. If you knock it out in mice, in neurons, they don't have any Syntaxin anymore. And then we can bring back the protein using a viral expression system. And then we can actually modify the Syntaxin molecule by putting mutations in it and sort of see how well this protein is now rescuing function. So that way we can really look at individual amino acids in the molecule, what it does to different steps in the fusion process. As I said, electron microscopy has become a very important technique. And so we have been using also, in particular, flash and freeze. It's a technique where you put your grown neuron in a high-pressure freezer, and then you stimulate the neuron either electrically or with an optogenetic stimulus, channelrhodopsin, to fire an action potential, and then a few milliseconds later you rapidly freeze the sample, and then you sort of arrest the synapse. You take a snapshot of your synapse structurally and analyze it then using electron microscopy. And then, for example, when you look at a typical picture or high-resolution picture of the presynapse, you see all these synaptic vesicles. You see the postsynaptic density. You see the synaptic cleft. And then you see some vesicles that are directly attached, some that are very close by, and some that are further away. And so we call them docked and tethered, and you can make this distinction. And then you can say, what happens if I induce an action potential? And you just basically simply count the number of docked vesicles along the presynaptic membrane. And typically you have like 1 or 2 of those vesicles along the profile here. And then you compare the number of docked vesicles after an action potential. You see the number drops by 30%. So that means we are, right after the action potential, we're losing these vesicles because they have successfully fused with a membrane. And essentially, we can measure with this the efficiency of the release process. So if you lose 30% of the vesicle, that means the per vesicle, the likelihood to fuse is, per profile is that 30%. So we're losing 30% of the docked vesicles with an action potential. And we can also see that they essentially recover over time. So this is kind of a refilling. Vesicles that have been depleted or consumed are being refilled by new docked vesicles. And the tethered vesicles do not change the numbers. So we see that only the docked vesicles are reduced. So we know that docked vesicles are actually fusion-competent vesicles from this experiment. This was an important link between docking and being primed and fusion-competent. Okay, so you can sort of, this is to illustrate that distance really matters a lot. And, this very close range between like 0 to 10 nanometer away from the membrane is very critical for a vesicle to be fusion competent or not fusion competent. And so we find that proteins like the snares or MUNC13 are very critical regulators of this distance. So if you look at the distribution of vesicle distance, so 0 is docked, and this would be a vesicle that's 100 nanometers away, and you just basically measure these distances, you see that typically for wild-type synapse you have quite a high number of docked vesicles, and it drops and then becomes kind of normal. And if you now make a mutation in the snare protein, for example, Syntaxin, you're losing the number of docked vesicles, right? So the ones that are directly attached are reduced. And so that means that Syntaxin is important for vesicle docking, okay? And here you see another example of MUNC13. MUNC13 is an important vesicle docking and priming factor. So if you remove it, you lose all your docked vesicles. And so the question is, what does it do? So according to the data before, we're losing vesicles that are docked with an action potential. So if you have no docked vesicles, what happens to release? Actually, you completely lose the ability to release vesicles. And this can be done, or for example, there's an experiment, not a genetic experiment, but like a clostridial toxin experiments like tetanus toxin or botulinus toxin. They are cleaving snare proteins. This is what they're famous for. And that's how we actually found that snares are important for fusion. These toxins are, are proteases of snare proteins. So if you either cut synaptobrevin or in this case SNAP25, you're losing synaptic responses completely and without affecting the firing of neurons. And you can see that in the case of cutting synaptobrevin, the vesicular snare, that all the docking, you don't have anything docked anymore, right? So this is a light blue line, and compared to black, which is a wild-type cell. So that clearly shows the snares are important for docking, okay? And obviously also for release. And there are some interesting things, like, for example, you can have you can have mutations in the snare protein, for example, in Syntaxin, which will be the major focus of my talk, where you are affecting the zippering of the snare complex. And in this case, there are certain layers that define the snare complex. I'll show you this later in a picture. And if you make these mutations, you also completely lose the ability to evoke response. So this is the response of a wild-type neuron. And here the evoked response is like 5 nanoamp in amplitude, and it's completely gone if you have no Syntaxin left. If you now bring wild-type Syntaxin back, you rescue the response. But if you now introduce a mutation in these positions, which are defined in these layers, you also lose the response completely. You also lose the ability to make fusion-competent vesicles, which is measured by the hypertonic response, and you also lose spontaneous activity. Okay, so you have a mutation, point mutation in the snare in Syntaxin, affecting the assembly or the zippering of the snare complex, and that has impairs all 3 forms of release in these neurons. And when you look at the electron microscopy, here is wild-type number of docked vesicles. This is in the knockout, so we're losing docked vesicles if you have no Syntaxin. And this is the docking brought back by the wild-type Syntaxin. But if you now introduce a mutant that has impairment of fusion, they still have normal docking. So we can see you can distinguish between docking phenotypes and priming phenotypes. So this mutant still docks perfectly fine, but is not capable of releasing. So there's something more than that you need. You need more than docking of vesicles. You need also some additional conformation of the snare complex that regulates release. And I'll show you some more of these examples later. So just to mention, docking and priming are both important, but docking doesn't mean that the vesicle is primed. Okay. So now let's go to the Syntaxin. Why are we working on Syntaxin? Syntaxin is one of the 3 snares, and it's a particularly interesting one because it has the transmembrane domain, of course, which anchors us to the plasma membrane. It has a snare motif which forms this zipper, this coil structure, and then it has this large N-terminal regulatory domain. And so because we were the ones that made the knockout of Syntaxin, we studied it up and down, looked at all the different domains, what do they do, okay? And so just to summarize what we've been doing so far, we found that the N-terminus is actually not super essential for fusion, but it's actually important for interacting with another molecule called MMP, which is also essential for fusion. So if you knock it out, you also lose fusion. But this HABC domain, these are kind of like substitute coil structures that can fold back onto the snare complex to stabilize it. And those, if you remove those, Syntaxin loses its function. And so that's kind of all published. And there is also a linker region here where sort of you can regulate how this region interacts with this region. And if you make mutations in here, you're affecting both the interaction between these 2 domains and also the interaction with MMP. And so you sort of can switch Syntaxin in open and a closed conformation with this and with this mutation, and that has also an impact on how it interacts with this MMP molecule. And so this will aid overall the fusion reaction. So what I want to tell you now about is actually some role of Syntaxin close to the membrane. So in fact, this kind of region here, basically this region, the transmembrane domain and the very proximal region of Syntaxin, how does it actually affect release? This is what I will be the first part of my talk. What does it do? Okay. The first thing that we were thinking of looking at is essentially we're looking at the transmembrane domain and the linker between the snare motif and the transmembrane domain. If the snare motif zippering is important for bringing distance, then of course the distance between the snare motif and the transmembrane domain must also play some role. So what we did, this region is not very long, it's like 6 amino acids long, we essentially added 3 additional amino acids, either at the edge between the linker and the transmembrane domain, or the linker and the snare motif, and sort of examined how insertion of these only 3 amino acids, what does it do to the release? And the effect was very profound. So if you actually, and was a bit surprising. So we first thought that inserting it between the transmembrane domain and the linker would have a strong phenotype, and that might be a weaker phenotype, but the opposite was true. So if you put the linker between the snare motif and the polybasic region here, this linker region, then we completely lose the synaptic response. This is a wild type. But we do have still some response left if we put the linker in here, and the response itself gets much slower. So we slow down the fusion process. It's still happening, and we can quantify it. It's pretty good, actually. Quite a lot of vesicles are released, but much, much slower. This is like the very basic release in wild type. This is a release in this mutant. So with this mutant, we slow down release. So obviously, this region is very, very sensitive to release, and it affects both the ability to fuse and also the kinetics of the fusion process. So obviously, this region is very important for fusion, and we're trying to understand this in a bit more detail. So the other thing that we noticed, even so we lost a major component of release, spontaneous release, the calcium-independent release, was majorly increased in particular in this mutant, indicating that this region has increased, not decreased, right? The other one, evoked release, was decreased, but we see now an increase of spontaneous release, arguing that there must be some kind of very sensitive regulation of these 2 types of release going here in really opposite directions with these mutants, which is kind of was very interesting. And so we said, okay, let's look at this region in more detail. And so we performed, essentially, these are only 6 amino acids here. We did some site-directed mutagenesis in this region and looked at various mutants and looked at evoked response and spontaneous release as the 2 major forms of release. And what we found was that mutating individual residues, and you can see most of them are actually basic. They carry a positive charge, which is interesting because the phospholipid membrane is negatively charged. So the idea is that there are some electrostatic interactions with this region, with the phospholipid membranes. And by single point mutants, we really don't see much of an effect on evoked responses, so they seem normal. But we have a massive decrease in spontaneous release, in particular if you mutate the position at the position 260, which is the first one, the one that is furthest away from the transmembrane domain. You would think that the ones that are close to the transmembrane domain have more interaction with phospholipids, but this is the furthest away, and this causes a change, a reduction in spontaneous release. You remember when we make a linker in here, then we get actually an increase in spontaneous release. So obviously there is something really important going on here for in regulation of spontaneous release, and so we investigated it further, basically just looking at evoked responses, at the pool, at the ability of vesicles to become primed, the probability of vesicular release, which is essentially simply the division, how many vesicles are released per action potential and how many vesicles are available. You know, I had this example of 300 vesicles are released and 6,000 are available. That would be a release probability of 5%. So that's this value here. And you see always a red dotted line is a wild type Syntaxin. And you can see that, for example, the individual point mutations have a small, close to significant effect on the overall evoked release. But the main impact was really on spontaneous release, and in particular, this position has we have a dramatic drop in spontaneous release activity. Okay, so then we didn't know what was the mechanism. Why are we seeing this change in spontaneous release activity? And then a classical experiment we do is, are our mutants expressed properly? So we always do Western blots and we do immunocytochemistry to see is a protein we express a virus actually expressed properly and properly targeted to the membrane. And what we discovered, and by the way, this is Gülçin Varda, who has really done most of this terrific work, in particular the first part. She found that depending on the position, this is a wild-type band of Syntaxin, depending on the position, we start seeing multiple bands. So this is a higher band than, for example, here, and the K260 mutation that had the major impact on spontaneous release run at a lower molecular weight than the wild type. And if you do the same experiment in HEK cells, you just don't see it. In HEK cells, they all run at a lower molecular weight. So there seems to be a neuron-specific increase of molecular weight happening with Syntaxin, and it requires these basic residues in this linker region. In particular, the position K260 seem to be very important. But you can see here there are 3 bands, right? So there's something interesting happening, and it smelled like post-translational modification is somehow modulated by these basic residues. So here, just again a correlation. We sort of had all these different band patterns, and we gave them like an arbitrary value. Like 6 would be like wild type, and 1 would be like the lowest one. And then we see that the lowest one has the strongest impact on spontaneous release, but none of them, the evoked response isn't really very well correlated. So that means if you're losing this molecular weight band to the lowest position, you have the lowest spontaneous release activity. Okay, so we have here a very nice correlation of a apparent putative post-translation modification to spontaneous release activity, which was kind of a surprising discovery, because that has not been, Syntaxin has been studied up and down over the last 30 years forever. This is the first time I've seen anybody finding some changes in post-translation modification. And so the question is what kind of post-translation modification it is. There are, in the transmembrane domain, 2 cysteines. And cysteines are often modified by lipid anchors. And in this case, it's palmitoylation. So you put a palmitoyl acid onto the cysteine. This is called palmitoylation. And we actually said, okay, if maybe this residue here regulates the ability of these cysteines to be palmitoylated. And indeed, so if you make single point mutants here or here, we see that the molecular weight drops slightly, and the double mutant drops even further, and this is mimicked by the point mutant here. And if you add these 2 mutants together, it doesn't drop any further. So arguing that the K260 and also the neighboring basic residues are essential for the palmitylation of these cysteines in the transmembrane domain. Okay, that was our hint. Why is this residue so important for regulating spontaneous release? It apparently is affecting post-translation modification. So to show this directly, there's a biochemical trick. You can take the purified protein from the neuron. You first block all the free SH sites from the cysteines, then you cleave off the thiol ester, so you remove endogenous palmitylation, and then you replace them with the biotin label, and then you use streptavidin to detect the sites that were previously palmitylated. And it looks a bit dirty, but it worked. So here, in wild type Syntaxin, you see this band, which corresponds to the palmitylated Syntaxin. The palmitylation is gone if you remove the cysteines in the transmembrane domain, but it's also gone if you mutate the position 260. So this confirmed very nicely that indeed we had here interfered with palmitylation with this mutation. And so what does the phenotype look like in these cysteine mutants? Do they actually mimic the phenotype of the basic residue mutant? Indeed, they mostly do. So again, we don't see much of an effect of an evoked responses, but we do see also suppression of spontaneous release, similar to the K260 mutant. Right? There's a reduction in spontaneous release activity. And again, it's not really additive, the effect. So clearly we seem to have found here a mechanism how spontaneous release activity is modulated by is it me or? I'm just reconnecting, let's see. Okay. All right. So, and that has also, but it's not exactly the same, because remember, these basic residues in the linker region can also interact with phospholipids. And so we do find that that this basic residue in position 260 has a dual role. On one hand, it enables the palmitoyl transferase to actually to do this post-translation modification. And then secondly, also regulates release probability. So this is not the only game that it's doing. So there is really, we're talking about some impact on release, which you can see very nicely. You have like trains of action potential. You look at the evoked response amplitude. So in wild type, you see this slight depression over time. And for example, in this mutant, actually the increases of time, which argues that the release probability is decreased in these mutants. So what I can say from this is that loss of palmitylation decreases vesicle fusogenicity in general. That's what we find. But the charge reversal of the K260 position, like making the positive charge negative, also affects pool size and increases spontaneous release rate. So basically, we can really, in detail, figure out what the molecule is doing, Syntaxin, how the Syntaxin molecule is actually performing its function. So you might ask, okay, what is this good for? Why are you studying this? Essentially, of course, we believe evolution has not for nothing created all these changes, putting a positive residue there, enabling palmitylation. The question is, how is it being actually used in the fusion process? And it's important to notice that not every synapse is alike. Right? The mossy fiber synapse is different than the climbing fiber synapse in the cerebellum. And the ribbon synapse in the retina is again different. And the hypothesis is that the fusion apparatus adapts to the needs of specific synaptic functions. Okay, and so and I will get to this in a second. Just to summarize what I just mentioned to you, showed to you, if you look at the linker region here in the snare complex, between the snare complex and the transmembrane, this residue here is important for this enzyme to put lipid anchors in the middle of the transmembrane domain. And you can imagine that these anchors actually affecting the structure and the behavior of this anchor, and probably also affect the fusion process itself. And what we found was when you make these mutations, you're affecting both spontaneous and also evoked response in a specific energetic manner. So that's what I've showed you so far. And so now I come to the question of synapse is not a synapse, is not a synapse. If you look at other synapses, like the most discrepant synapse in the brain is a ribbon synapse, because there you don't have action potential induced release. It's like membrane potential induced release, and you have like chronically releasing glutamate in the photoreceptors in the bipolar cell. And so you can see that in these chronically releasing synapses, you always have Syntaxin 3B instead of Syntaxin 1A. So there is really a switch between different Syntaxin isoforms. There is no Syntaxin 1A in the ribbon synapse, but there are Syntaxin 1A in the chronically, phasically releasing synapses, for example, between the bipolar cell and the ganglion cells. Oh, okay. So what has it to do with this? Now, interesting thing is, if you look at the sequence between Syntaxin 1A and 3B, they're actually quite similar. They all have a transmembrane domain, they all have a linker, they all have a snare motif, they all have a regulatory N-terminus. And look at this, at the position which we identified to play such an important role in spontaneous release, this K260E, is actually an E in the Syntaxin 3B. So the mutation that we used is actually used by nature in a different in the ribbon synapse. Okay, so we said, okay, so how does Syntaxin 3B works in a central synapse? We didn't do the experiment around. We didn't have a chance to look at the ribbon synapse because it's also a difficult synapse to work with. We lot of simple synapses to work with. So we stick with our hippocampal synapse, and we put the Syntaxin 3B in our knockout background and see what it does. So first of all, we find that the Syntaxin 3B can express in neurons and goes to the synapse. And you can also see that the band increases if you put, instead of the E, a K. We sort of put the basic residue back into the Syntaxin 3B backbone, like B like Syntaxin 1A, and suddenly the band size increases, presumably because we reinstate cysteine-mediated palmitoylation in Syntaxin 3B. Okay? We know in Syntaxin 1 this has to be a K to get this palmitoylation. Here is an E, so it should be blocked. And now we put a K into in 3B, and boom, we get an increase in molecular weight. So we're enabling palmitoylation. So what does it do to synaptic transmission? There is absolutely no synaptic transmission in the wild-type Syntaxin 3B, but we can rescue it by putting back the basic residue and enabling palmitoylation. So for Syntaxin 3B, in this hippocampus context, you must have a basic residue there, and probably you also must have palmitoylation in this region. So arguing that this is actually very important for certain fusion function, at least at the phasically releasing synapse. And the chronically releasing ribbon synapse, probably the mechanism is different. And we can see here that this has actually been used also by nature to modify palmitoylation activity depending on which syntax and isoform is expressed in which synapse. And therefore, you can tune synapses to be phasically releasing or even chronically releasing in terms of the ribbon synapse. Okay, so, and the interesting thing is also spontaneous release is also rescued. In fact, we see more spontaneous release in this mute in Syntaxin 3B than in Syntaxin 1. And we'll get to this in a second. So now I come to Syntaxin 3A. This is a splice variant of Syntaxin 3B. And Syntaxin 3A is very important in postsynaptic neuron function. For example, secretion of AMPA receptors in LTP requires Syntaxin 3A function. And so the hypothesis that snares are not only important for the presynapse, but also for postsynaptic vesicle fusion by enabling vesicles that contain AMPA receptors to be brought to the plasma membrane. So if you compare the sequence of Syntaxin 3A with Syntaxin 3B and Syntaxin 1, what you find is they do have a Q here instead of an E. So it's a more neutral residue, and they're lacking the cysteines here. And so we can again reintroduce palmitoylation by making chimeric proteins between Syntaxin 3A and Syntaxin 1A, and we can reintroduce the palmitoylation, which is a lower band, by putting both the transmembrane domain of Syntaxin 1 and the linker region of Syntaxin 1 into the molecule, and then we can make Syntaxin 3A again being palmitoylated. And how does a phenotype look like? So Syntaxin rescue of Syntaxin knockouts, 1 knockouts with Syntaxin 3, you get a little bit of release, and the release is actually slower than Syntaxin 1. And so it's weaker, but the other interesting, and you can see here in the time course, it's also the decay rate takes longer, meaning that the fusion is a more slow process. And but what is interesting is even so we can rescue this activity by putting back mainly, the Syntaxin 1 domains. One thing that is still remaining is a strong increase in spontaneous release activity. So we rescue evoked responses largely, but we suddenly, in the Syntaxin 3A background, we suddenly see more spontaneous release activity than with Syntaxin 1, which argues that Syntaxin 1 may be specifically designed to have low spontaneous release activity. Okay, so and that is when you look at this difference in sequence between Syntaxin 3b and 3a, these are actually splice variants, okay? So, Syntaxin 3 and Syntaxin 1 are different genes, but 3a and 3b are splice variants, and the splicing happens in the last part of the snare complex, exactly from the center of the snare complex to the end. And when we now replace the entire C-terminal portion, we gain back evoked responses, but we still have enhanced spontaneous release activity compared to Syntaxin 1, which argues that this region here, and also compared to 3b, which argues that this region here, which is part of the snare motif, contains sequences that in Syntaxin 1 suppress spontaneous activity and enhance spontaneous activity if you have Syntaxin 3a there. So we have now, using this chimeric approach, we have now identified another region in the Syntaxin regulating spontaneous release activity. Okay, this is kind of what we want to know. We want to know how does spontaneous release activity occurs, how does evoked release occurs, and we identified that this region seemed to play an important role in Syntaxin 1, suppressing spontaneous release. We can discuss later why it is good to suppress spontaneous release. Maybe at the end we can talk about the physiological importance of this process. Okay, so Andrea joined the lab as a graduate student, and teaming up with Gülçin, who is doing the work so far. And so she looked at Syntaxin 2, another isoform. And here we found that the phenotype was even stronger. So we found even a more slowing of release versus Syntaxin 1. And we also found that vesicle priming was impaired, but we found a drastic increase in spontaneous release activity. So we have here another Syntaxin. There's a big family of Syntaxins playing a role for vesicle trafficking in cells. But this one is very bad in actually triggering evoked responses, but very good in triggering spontaneous release, right? Spontaneous release is kind of, you can think of it as a background noise. You know, they just don't, they fuse without even having a stimulus, which is not really a good thing, right? You want to have a specific signal linked to the action potential firing. So this Syntaxin 2 is really promiscuous fusion, a snare molecule that enables promiscuous fusion without involving a trigger. And so we said, let's look at this Syntaxin 2 because that's such a strong phenotype to understand the role, the domain activity of spontaneous release. We already know that the C-terminal portion of the snare complex from the comparison of Syntaxin 3a versus 1 is important in regulating spontaneous release activity. How about Syntaxin 2? Just sort of look at it from another molecule. And so Andrea knows when she compared the full-length Syntaxin 1 with the full-length Syntaxin 2, we have big increases in mini activity. She made now chimeric proteins between 1a and 2 and sort of replaced sequence domains of 1a with 2. And she found again that the evoked response wasn't really much affected. But again, she found a massive increase in spontaneous release in particular when the C-terminal portion of the snare complex was replaced with Syntaxin 2. Again, similar to the Syntaxin 3a, this region here seemed to be very important for regulating spontaneous release in Syntaxin 1. Okay? So this is a bottom message from this. And she did also the inverse mutation, putting Syntaxin 1 sequences into 2. It's probably a bit complicated, but again, it points that the snare motif, and particularly the C-terminal portion of the snare motif, are very important for phasic triggered calcium release. And and you can see it here also very nicely. If I put the Syntaxin 1a sequence into the Syntaxin 2 backbone, we massively drop the spontaneous release activity to levels that are typically for Syntaxin 1. So this is a release activity of Syntaxin 2. This is Syntaxin 1. You see a big drop. And we can just simply exchange this region of Syntaxin 1 into Syntaxin 2, and that drops spontaneous release activity. Okay? So obviously this is very important for suppressing spontaneous release. And so what's going on there specifically? Now, there are a couple hypotheses. One hypothesis we found was actually that recruitment of this other protein that I mentioned, MUNC18, which is very important, it's kind of a partner of Syntaxin. So Syntaxin 1 and MUNC18 together go to the synapse, sort of prepare the snare complex for forming, okay? And if you disrupt this interaction, you often have a disruption of synaptic transmission. So what we found was that Syntaxin 2, instead of Syntaxin 1, is much worse in recruiting MUNC18 to the synapse, okay? So, and we found that the ability of the Syntaxin to recruit MUNC18 to the synapse correlated very nicely with the suppression of spontaneous release activity, as if it is important for MUNC18 to be there to have the specific function. You can see it here nicely. This is the Syntaxin knockout. We have very little synapse. This is like a dendrite. This is a dendrite, and these spots are synapses. You see there's much less MUNC18 present than in wild type. In Syntaxin 2, we have very little MUNC18, but it comes back. Actually, if we put the snare complex of Syntaxin 1 or the C-terminal portion of the snare complex onto Syntaxin 2, so we now start recruiting MUNC18 to the synapse, and we again have a major impact on synaptic transmission. So it looks like that the importance of this region is, one important thing of this region is to recruit this cofactor MUNC18 to the synapse. And here it's kind of like, we like these kind of plots which summarize our data always very well in mutants. So the black spot is like the behavior, we correlate evoked responses to MUNC18 presence. This is the normalized to Syntaxin 2, which is low, and in wild type is around 1.7-fold higher, the level. And you also see that the evoked response is much bigger. And the blue spot is the Syntaxin 2. You see very little release and very little MUNC18. And for example, if you look at, for example, at spontaneous release activity, we see that both the Syntaxin 1 and the Syntaxin 2 with a C-terminus of Syntaxin 1 is completely suppressing spontaneous activity. It's behaving like wild type. While the Syntaxin 2 is really bad in recruiting MUNC18, it has a very high mini-frequency. And interesting, if you replace the entire snare complex, it's kind of intermediate. So we lose a little bit of MUNC18 recruitment, and we also cannot really fully suppress spontaneous release activity. It's actually much better to only replace the C-terminal portion. So this means that it's a bit complicated. This means that the entire snare complex domain somehow bidirectionally modifies MUNC18 recruitment and therefore makes a complicated phenotype, let's say it this way. You can see also very nicely that, well, I think let's stick with this because it's anyway already pretty complicated. But we kind of use these plots to really understand what these interactions do to the fusion apparatus itself. So, as I mentioned, we know now that the C-terminal portion of the snare complex is really important for spontaneous release, for priming, for evoked release, for the speed of release. So let's look at this more closely. And so we just compare between Syntaxin 2 and 1 which residues are actually different. And there are not that many because they're quite conserved. And so that allows us now to go for single amino acids. And sort of, this is work in progress. And Andrea is sort of going through one residue after another, single point mutations, and essentially plotting its effect on release, evoked release, on spontaneous release, on pool size, and on release probability. And the key thing for us is to screen now for single sites. We want to see atomic changes affecting release, spontaneous release. We want to look at some phenotypes where there's no change in the number of fusion-competent vesicles. The pool size should not be changed, but we want to see an increase in spontaneous release activity. So here you see Syntaxin 1, here you see Syntaxin 2, and here you see the increase of spontaneous release when we replace the entire C-terminus with Syntaxin 2, high in mini-frequency. So now these are single point or double point mutants. So for example, mutating the position 231 actually increases spontaneous release activity, not quite as exchanging the entire C-terminus, but quite a lot, without affecting the pool size. So that seemed to be an important residue for the disinhibition of spontaneous release. Meaning, if you mutate this, you're losing the suppression of spontaneous release. Okay, so we're looking for a couple other ones. So in particular 231 seemed to be important, 232, and maybe also the site 249, which is actually a serine. If you make it to glutamate, actually you also get an increase of spontaneous release activity, which is interesting. You could mimic that by phosphorylation of that site. So we will actually, this is brand new, I got this data today, this morning. Maybe we are now looking if this site actually might be phosphorylated, because that would be a way of switching the snare motif, snare complex between different modes. Actually, you see also it increases release probability. So it would be a way of getting like an LTP-like effect, just simply by phosphorylating this site. Anyway, so we are sort of scanning this through, and just to end this, brings another hypothesis. One hypothesis mechanistically I already told you about is this recruitment of MMP-18. The other idea is, it's just very famous, two Nature papers from Süthoff and Axel Brunger, showing that the snare complex interacts with synaptotagmin. Synaptotagmin is this famous calcium sensing protein. And they said this interaction is actually important for the triggering of calcium triggered release. We doubt this, we don't believe this. I mean, I'm a friend of Tom Süthoff, but we disagree on this very, very much. We think that this interaction may actually play a role in regulating spontaneous release. Because look, these two residues we actually identified in the past are very important for fast calcium triggered release. In their crystal structure interact with this position 231, which I basically just showed you is when you mutate this, we increase spontaneous release. Okay? So the hypothesis would be, when we mutate this residue or this, essentially we're disrupting this interaction with synaptotagmin, and that unclamps spontaneous release. Synaptotagmin not only is important for fast calcium triggered release, it's also important for clamping spontaneous release activity. That's another function that is that is known for. And indeed, when we make this mutation, you see this increase of spontaneous release activity just by essentially making mutations that disrupt this putative interface. We don't know if this what this crystal structure is. They found the crystal structure, but what does this crystal structure mean? What does it reflect? In which part of the release process does that play a role? So we think that might play a role in clamping spontaneous release activity. And that might be the thing that we have identified, the C-terminal portion of the snare complex, as I told you, is important for suppressing spontaneous release activity. And this might be by enabling the interaction with synaptotagmin. Okay? So, and this data is consistent with this. However, I must say, I find the MMP-18 recruitment concept, hypothesis, almost equally possible. So we don't have proof therefore yet for this whole thing yet, but I think both things are still into play. But you know, they cut two Nature papers out of this hypothesis, which not really proven yet, I must say. Okay. Anyway, I'm coming to the end. So what I showed you in the second part of my talk, by looking at Syntaxin paralog analysis, we found that the different, by using the Syntaxin isoforms, we can show regulation of efficiency, speed of release, priming, and also spontaneous release is regulated, can be sort of dissected apart on an atomic level. I also showed you that recruitment of MMP-18-1 to the C-terminal portion of the Syntaxin snare motif is really important for what we observe, and can serve as an alternative mechanism in this regulation of spontaneous release to the idea that synaptotagmin interaction to this region may do that job. Maybe they do it together. I mean, it's possible, right? Maybe they switch with each other. Who knows? Anyway, so that's kind of the the end of this. Most of the work, the first part is published recently. The second part with Syntaxin 2 is not published yet. So don't distribute this to the net yet. But of course, I present the data, so it's common knowledge. So really, Gulcin Vada, postdoc, she was starting as a graduate student lab and then continued as a postdoc. She really did a very independent, very thorough analysis. She, worked for many years on this palmitoylation mechanism and this the mutagenesis. And Andrea is sort of stepping into her legacy essentially by looking at the Syntaxin 2 mutants. Xu Wen Zhang and has been, I only briefly showed the data, very important for understanding synaptotagmin, role in docking and priming and maybe and suppressing of spontaneous release. And Verrechte was doing quite a lot of EM for us, and Thorsten made all the mutants and the viruses that we use in this study. And Shigeki Watanabe and Eric Jorgensen came to my lab in a sabbatical, and we together developed this flash and freeze in central neurons, and this really also transformed my lab dramatically, because it's a really cool technique that you can take snapshots with millisecond precision on synapses, when in sync to the action potential. And yeah, this was mainly supported by the German Foundation for Research, DFG. And thank you for your attention. I know lots of detailed stuff. This is what we do, mechanistic stuff, and not big picture and culture, not in vivo behavior. So we're really kind of mechanistic-y guys working. And I'm glad you still stick somewhat with me along this data, and I'm happy to take questions.
Okay, I think now you believe me why I'm not working on this synthetic transmission, because there are so many smart guys, including Sudhoff, and also so many famous people. Yeah. So now let's open for question. And because I think it's very complicated, so maybe we start from some simple questions. So maybe I start asking some technical questions, sure. Technical issues. So I'm also very interested in this new technique. It's called fresh and freeze. Yeah. Mm-hmm. And when I visit Peter, he also showed me this interesting toy. Yeah. Because he collaborated with Shikimoto. Yeah. You show that technique, and you can have this ducking. So ducking is you say is a kind of an anatomical term, right? Yes. You can count the number of the ducking vesicle. Yeah. But before this technique, we always use this electrophysiology approach to measure the readily released pool, and then we can extrapolate to tell people what's the readily released pool. Yeah. And I also found that throughout your presentation, you show a lot of this readily released pool, what's the number. Yeah. So what's the correlate between these morphological, this duct, and also this functional term, this readily, are they exactly the same, or there is a difference?
So I think this is very important, and that's of course very central to our work, trying to understand. And I think this piece of data is very important because it does, what we do is we deplete, we consume vesicles, and we find a reduction of a specific population of vesicles, and only the docked vesicles are consumed. Right? Mm. And then you have like a protein like MUNC13, famous protein. You only see these tethered vesicles. They never touch the membrane. They all look like this, never like this. Mm-hmm. And they can never, they don't dock, and they never fuse. Mm-hmm. So that argues, also on the molecular terms, that there's a very nice correlation between docking and fusion competence. Okay, mm-hmm. So if they can't reach the membrane, they cannot fuse. So it's essential that they get there. But I showed you in the other slide the Syntaxin AV mutant, and also here, no, this one here. This mutant here has absolutely no release, right? There's no release happening, no priming, There's no pull. There's no spontaneous release activity. Right? It's flat, and yet they still dock completely normal. Okay, so that means docking is necessary, but it's not as sufficient to enable fusion competence. Okay, that's the important thing. So there are really two steps. So you must have docking, but in addition you must have also a functional snare complex. Essentially, let's call it this way. Okay, that would be the best description. And if you make mutations in the layer of the snare complex, and I'll show you where that is. So this is like the snare complex, and here is the zero layer. This is the middle. This is called the zero layer, and the residues that are sort of in the coil are layered in between -7 and +7. Okay, they are like 15 layers, and if you make mutations here in the layer 3, layer 4 region, you completely block synaptic transmission, because probably they zipper up until to this point, and then after that they cannot fully zipper anymore, and then you block. And they might still dock. This might be compatible with docking. So in EM you cannot distinguish between a vesicle that is partially zippered and fully zippered, right? That the EM technology doesn't allow you to do that. But essentially what it means is zippering up all the way to plus layer 7 is important for being a primed vesicle, a fusion competent vesicle. If you make mutations that disrupt this, you don't get fusion anymore. And I think also the botulinus toxin A affect the one that is injected in the skin, right? That one also cleaves SNAP25 just until this piece of SNAP25, and therefore it prevents the fully zippering of the snare complex. And again, you have no fusion.
And also, one question is, there are so many isoforms of Syntaxin.
Yes.
Some influence physical release or spontaneous release. Yes. So under physiological conditions, how different Syntaxin coordinated together for this complicated synaptic transmission?
Yeah.
So
my view on this is, as I said, there's a high diversity of synapses in the brain. Like you have tonically firing, you have spacically firing, you have high probability, low probability synaptic release, and I think essentially like neurons can fire very different patterns of action potentials. You co-evolve the release apparatus to the firing behavior of the neuron, and the connection, the meaning of the connection. Do you want to have a strong connection or a weak connection? Therefore, you rebuild, you vary the dialect the synapses speak in by varying the protein content, by putting different Syntaxin isoforms, different splice variants, different...
In one single type of neuron, they can express different...
In principle, in a single type of neuron could be, but I think generally I would say in this case, in this extreme case, I can say certainly ribbon synapse has different snares than the hippocampal synapse. And that makes a lot of sense, and probably also, the inhibitory neurons use they use a different synaptotagmin, and they have slightly different components of the release machinery than the excitatory synapses. So we are very interested in this diversity of proteins of the presynapse shaping the behavior of synapses. And you can also change it in plasticity or development. Let's say in the early time of the development, maybe you want to have high probability of release, so the synapses find each other, and later you want to scale down release probability, and you're putting Syntaxin 1A in there and not 1B, or you put the MUNC13-1 in there, not Syntaxin 2, MUNC13-2, and so on. So I think gene expression regulation is going to play an important role in shaping synaptic behavior. Yeah.
So finally, sorry, I have the last question. So speaking of this synaptic plasticity, so as in electrophysiology, we often induce LTP, and then we check the pair pass ratio, and that's the physical release.
Yeah.
But sometimes we also check spontaneous release,
yeah.
and we found this miniature EPSC or IPSC is increased,
yeah.
and we interpret this as LTP after some kind of behavioral paradigm, and we found, we added the TDX and we found the miniature EPC is increased. But from your talk, actually, there's two different processes. And what happened to this Syntaxin in this case?
Exactly. So it's possible that if you look at the frequency of minis, that in fact is accompanied by a decrease in the evoked responses, not an increase. So it's possible. You can have a diversion of these two phenotypes. Now, I want to say that in many cases, when we see an increase of mini release, we also see an increase in evoked release.
Exactly, yeah.
This is a general increase in vesicle fusogenicity. So the vesicles become more prone to fuse when you stimulate them.
Yeah.
So, for example, if you apply phoboesters, a classical manipulation, actually that binds to MUNC13, it makes the vesicles more fusogenic, and you see both an increase in the evoked response and both an increase in spontaneous release. Or let's say you add complexin to the snare complex. Complexin is the best studied snare complex binding protein. It's a short peptide, like 130 amino acids, and that actually has multiple functions depending on the synapse. Very interesting. But in the central synapse, it promotes both spontaneous release and evoked release. In the fly neuromuscular junction, different setting, right? Neuromuscular junction doesn't have inhibition, right? There, it is mainly there to suppress spontaneous release and not so much in increasing spontaneous release. So you make slight changes to the structure of these proteins, and then you twist the release machinery to behave in a slightly different way. But it's basically telling us that nature has used a toolbox of molecules to tune synapses to behave in a very specific fashion.
Okay.
And yes, go ahead.
I'm very curious about the palmatolylation, because you said only the first chart is a lysine, right? Or the
it's K. Yes,
it's a K, right? So why only the first lysine is very crucial? Because if there's a stretch of charged residues, right?
Yes. So it's not really the first one. You can see actually multiple. They play. So here we go. Here you can see it actually quite nicely. So you can see this is the K260 mutant, right? This is the Y type. This is the kind of no palmatylation phenotype. But you see already a little bit of a higher band here.
Okay.
And this mutant, this site doesn't affect palmatylation, but this site, 2 amino acids further, also have an impaired palmatylation. And this one again has another pattern. And this has mainly the middle band is strong, but not the. So really, you see, depending on the amino acids, 1 or 2 or non-palmatylation thing. So that means the entire region really affects the binding probably of the palmityltransferase. Enabling then the modification of the cysteines in the membrane. So, but this has the strongest phenotype, and as I showed you here, the correlation of the single point mutants, right? If you could combine them, you would see correlation of spontaneous release and the palmatylation is, the strongest effect you see with the position 260. But for example, this one also, this is I guess 262. Also has quite a strong effect, while this right next to it has no effect. Right? So it's just it's a region, and there is some specific areas within the region that are particularly important.
So the structure of that region is solved, or is
it's helical. I mean, I think that's what people know. And the orientation, also the question is syntaxin, they can form bundles, maybe there are multiple syntaxins neighboring each other. It's not really well understood. Yeah.
Last question. I'm just very curious whether there's any non-human mutation that's associated.
So in this region we don't have, but we have actually studied mutations. There's another paper we have published that looked at syntaxin mutants associated with epilepsy. And the mutants are either in the snare complex or in the N-terminal portion of the molecule. It remains to be I'm sure we will find more mutants over time. This is like exploding. The human genetics field is for us one of the most important resources for mechanistic ideas, because you know you have these 8 billion animals, highly phenotyped, right? The human population, right? You can see what there is wrong. You can sequence them, and then you can see, okay, MUNC13 has a mutation on this side that causes epilepsy. Let's see what this does to the function of the molecule, right? So we are actually using these human data to understand the mechanism of the fusion apparatus itself, because we know that it somehow interferes with function, right? So it's a wonderful interaction with clinic and mechanism that we actually do in the lab a lot. So I have a lot of students coming to my lab now. We use also human iPS cells in our work now. We make mutations in synaptotagmin and snares and human iPS cells and make neurons out of them and then study the phenotype. Right? Lots of students are interested in this question because they're trying to understand disease, you know, and this is actually, you can directly go from the patient, make a stem cell, divide them to neurons, and then see what's wrong with my synapse, right? Why does this patient have epilepsy or learning disabilities and so on, or being schizophrenic or whatever. Right? Lots of potential, but still hard work.
So when we learn about synapses, there are like this analog synapses, like the ribbon synapse that transmit analog information, and then the central synapse that transmits the digital. And it was very interesting to know that different syntaxin isoform really like perform different things and optimize for evoke and spontaneous release. So, and it's also interesting to know that like the spontaneous release is not completely gone, and also so what do you think is the function of those spontaneously releasing central neuron? Does it really?
Thank you for asking that question.
Yeah.
Because the simplest view is, once a vesicle is fusion competent, so you sample the release machinery, you get this snare complex is zippered up, you have MUNC13 in place, everything's in place, and it's just waiting for the calcium signal to come, right? So the vesicle is already very close to the plasma membrane, and it waits for this trigger. And the energy barrier for fusion cannot be too high at that point, because all we add a few calcium ions to the system, right? There's very little energy available. So you have to lower the energy barrier quite a lot for fusion to take place. And by lowering this energy barrier, you automatically allow spontaneous fusion to occur. Basically the difference between spontaneous and calcium-triggered release is a factor of 50,000 approximately, depending on the synapse. So it's a byproduct. Spontaneous release is automatically a byproduct for making vesicles fusion competent. Now, this can be bad because think about it. We have, as I mentioned, I have 6,000 fusion competent vesicles in this neuron, right? So if each vesicle fuses once a minute, that we would have 100 vesicles per second fusing. Lots of background noise, right? So what you want is you want to suppress spontaneous release as much as possible and enhance calcium-triggered release. You want to separate these two forms of release as much as energetically as much as possible. So therefore, fusion clamps like synaptotagmin or complexin can be very, very useful to make the signal to noise in the synapse better. Okay, so in a sense, spontaneous release is something bad in the central nervous system setting, because you have 10,000 synapses onto one neuron, right? And if each synapse releases a vesicle per minute, you have like so many spontaneous background noise that you can't really see the activity anymore. The good thing about the central nervous system is we have both inhibitory and excitatory synapses at the same time. So if they both release spontaneously, they sort of level each other out, right? The problem in the neuromuscular junction is that you don't have that. The neuromuscular junction has only an excitatory input. And that's why you suppress there spontaneous release much more, because you don't want the muscle to twitch spontaneously, right? You want the muscle to only fire when there's an action potential coming.
So is it possible that it's not
completely noise? Like, could that be a remnant of analog signaling in the...
Yeah, you could think of it, one way of thinking is you're increasing the conductance of the neuron by spontaneous release. Yeah, or you could create a variation memory potential which helps the neuron to fire. Yeah, but then you would have to have a mechanism that modulates spontaneous release activity also, right? Like a phoboester-like response, right? Phospholipase C activity would be one idea, but phospholipase C activation is something that happens mostly at the postsynapse. There's a G gamma, GQ, G protein Q. It's a postsynapse, and that activates phospholipase C, makes DAG, and DAG is the phoboester analog. So if you tell me a G protein coupled receptor at the presynapse that stimulates phospholipase C, I give you $100, okay? Because this would be very cool way of modulating presynaptic release by affecting spontaneous but also evoked release. But all we know is all the G protein coupled receptors all couple negatively to release by blocking calcium channels and activating potassium channels presynaptically. Yeah.
So very nice talk. So quick question. So the linker region, so you talk about the palmitoylation, right? But the snail protein need a codomain and a linker region. I'm wondering whether linker region enough palmitoylation, will that affect the protein interaction, such as the snail protein interact with MUC18 or synaptotegment?
Yeah, so palmitylation, as I said, occurs in the transmembrane domain. So you would first think that wherever that it could be protein transmembrane domain interaction. This would be the first thing to think of. Let's say, with another transmembrane protein might be something. People have argued maybe that calcium channels might actually be important. The interaction of this, and this is really interesting, right? Intriguing idea. What you want for a release apparatus to work very efficiently is to have the fusion competent vesicle right next to the calcium channel, right? So the early work on Syntaxin has focused on actually seeing that it affects the function of calcium channels. So people have argued it would affect gating of calcium channels, and this would argue also direct molecular interaction. In fact, we have a little bit of data that suggests this, that there is an interaction of Syntaxin and to calcium channel function, but it's very weak. And this is like the old hypothesis that you could affect the key transducer in the presynapse, the calcium channel, right, which is a triggering signal, right? When that fires, then you get calcium influx and it causes freeze. But other things I'm not aware of because it is in the membrane. Of course, putting a lipid anchor right in the middle of the membrane will affect the local stability of the membrane very much. So we think that this modification makes the membrane maybe less stable and therefore more fusogenic, right? So you want to overcome the energy barrier. You want to break down two membranes into one. That means overcoming high energy barrier. And so if your membrane is initially locally unstable, by putting this anchor in there, it may be much easier to get fusion happening. That's one thing that where you think this palmitylation in particular is particularly useful. Yeah.
Okay, so any questions from students? You can ask any kind of naive questions. Like
Yeah, please.
Okay, so I think it's also this time.
Yeah.
Thank you. Yeah. Let's thank Christian for his wonderful talk again.
Thank you for your patience.
And so we have some souvenir for you.
Oh my God!
So this is, I think this is on the radio. It's not a lot of money. But
No. Thank you so much. I didn't expect this.
Send
me the copy. Yes.
And also
Thank you.
we have this just for.
Wow! Look at this.
This is more interesting for you. So this is certificate. You cannot read it. So,
okay.
But you can keep this.
Thank you so much.
Thank you. Maybe we can take a picture. Yeah. Thank you.